Okay, Holly, we're good to go.
Okay. Thank you for attending tonight, everyone. Tonight, we're going to talk about COVID-19 vaccines and hopefully provide you with some information on the vaccines in general and give you some more knowledge on how to make choices related to your vaccine and what it looks like after you're vaccinated. So I am Nurse Holly, and I'll be your tour guide tonight. So basic backstory, COVID-19, everyone's very familiar with. There are actually 7 types of coronavirus that can affect humans. The viruses all share a similar shape. As you can see, they're round with a spiky surface, and the surface resembles a crown, where you get the coronavirus from. The coronaviruses can range from mild to severe. Mild forms would be the common cold. More severe types have been seen throughout the years. In 2003, there was the severe acute respiratory syndrome. And in 2012, there was the Middle East respiratory syndrome. Those were both more severe SARS-CoV-2 viruses. And now we have COVID-19. Evidence suggests that all 3 of these particular COVID viruses originated from bats and spread to humans. Whoops. Let me go back a little bit. There we go. So initially with COVID-19, there was a specific list of symptoms that we used to identify potential cases. As the illness has developed and we've had a year to look at cases, we see that there are a multitude of symptoms attributed to a COVID infection, including but not limited to cough, congestion or runny nose, body aches, fatigue, headache, and loss of taste and smell. Some additional symptoms may include a fever, nausea, vomiting, or diarrhea. COVID-19 is most commonly spread through respiratory droplets that are expelled when a person talks, coughs, sneezes, or sings. And these droplets enter the air and they can be inhaled by anyone in close range. Persons exposed to COVID may develop symptoms anywhere from 2 to 14 days after exposure, but typically we see symptoms develop around day 5. Once infected with COVID, a person is considered contagious 48 hours prior to the development of their symptoms. The severity of illness varies across the population. There are cases where people are completely asymptomatic and don't know that they had the infection at all. And then we have cases that present as a cold or a stomach virus. And then the most severe cases would be hospitalization, ventilation, and death. Cases in ages 65 and up continue to have the highest death rate related to COVID-19. And in Chester County, the 20 to 29 age group represents the largest number of cases of COVID-19, followed by ages 50 to 59. So when we look at the 3 scenarios of a COVID infection, we break it down to person A, person B, and person C. So person A is the person who's actively infected with COVID-19. Person A is contagious, and anyone who comes in contact with this person less than 6 feet for more than 15 minutes is considered exposed to COVID, making them person B. Person B is not currently considered contagious, but does need to quarantine for 14 days from exposure to ensure that symptoms don't develop. Person C did not have contact with person A, but may have had contact with person B. Person C does not need to isolate or quarantine at this time, as they were not directly exposed to a COVID case. You may experience this scenario if you or someone in your home were exposed to COVID and needed to quarantine, yet the rest of your household didn't. In this household situation, if person B, the person quarantining, were to become ill, they would then become person A, and anyone in the house who was person C then moves to person B and needs to quarantine. So these are the criteria that we use when we are notified of a case, when we do contact tracing, and then when we advise people to isolate or quarantine. So like I said, we do advise individuals who are sick with COVID-19 regardless of their symptoms, to isolate, and those who are person B, who are contacts, to quarantine. So the difference between isolation and quarantine is shown on the slide. Isolation is a 10-day period from when symptoms begin or a COVID test is administered in someone with symptoms. Quarantine is a 14-day period that begins after last contact with the COVID case. So the last contact is day 0. So the quarantine begins day 1, the next day, and then runs the 14 days. So yesterday we received some good news related to vaccination and some new CDC guidelines. These guidelines were updated and applied to fully vaccinated individuals. A person's considered fully vaccinated 2 weeks after their second dose of a 2-dose vaccine or 2 weeks after their single-dose vaccine. Once fully vaccinated, individuals may now gather indoors with other fully vaccinated people without a mask and or with unvaccinated people from one other household without a mask, unless there are high-risk individuals in that household. The requirements to quarantine or test after exposure have been eliminated by the CDC unless the exposed individual begins to display symptoms of illness. So if you go back a few slides, if you were person B and you're fully vaccinated, you would not need to quarantine or test for COVID. However, if you were to start to display symptoms of illness, you would then want to start to quarantine and get tested. The CDC still recommends masking and social distancing in public places and when gathering with multiple households where not everyone is fully vaccinated. The not so great news is the CDC is not a governing body, so these guidelines don't automatically become the law of the land. And right now, as of right now, the Pennsylvania Department of Health has not adopted these newly released guidelines. Typically, states do fall in line with CDC guidelines a few weeks later, and then it kind of trickles down from state to local government. So hopefully we will see some of these guidelines get into Chester County within the next few weeks. This slide is just an example of how the efforts that we do right now at school with our mitigation prevents the spread of COVID-19. Every layer of mitigation that we use, as far as the masks, the distancing, cleaning, and hand washing, creates a situation where it's harder for the virus to spread. Until vaccines are widely available, this is really the best method for us to prevent the spread of COVID in our school. We recently were able to add another layer of mitigation, and that is in the form of COVID testing for staff and students who come to school feeling well, but start to become sick during the school day. So with the COVID testing in school and outside of school, there are molecular and antigen testing kits, and they use secretions for the nose or mouth to test for active COVID infection. The test results may take minutes to days, depending on what type of test you have administered. Any test is subject to error, but the molecular testing is the most accurate tool that we have in testing for COVID. A person who displays symptoms of COVID and receives a negative rapid or antigen test should then receive a follow-up molecular test to rule out a false negative result. Antibody tests used with a blood draw look for antibodies which would indicate a previous COVID infection. Advances in testing continue, and now some at-home tests are available to the public. At Avon Grove Charter School, we use the Binax Now antigen test in our nursing offices. And like I said, we do, with consent, test staff and students who become sick during the school day with symptoms consistent with COVID-19. The Binax Now testing does not require a deep dive into the nose. So if you've had COVID testing done, like this poor fellow on the slide, we don't have to go up and touch your brain. We simply just go up just about a quarter of an inch, and we take a sample from both nostrils. the in-school testing that we have is available through Project ACEIT, which is an initiative supported by CHOP and the Chester County Health Department. So like I said, by having these tests at school and in combination with our nursing assessments, we can identify students who aren't feeling well, test them for COVID, and then should they test positive, we can begin to the isolation and the contact tracing process right away. So it's a real time saver and really prevents illness from spreading. So on to vaccines. Right now we have 3 vaccines that are being administered under an emergency use authorization through the Food and Drug Administration: Pfizer, Moderna, and Johnson & Johnson. Pfizer and Moderna are similar vaccines, and then the Johnson & Johnson one is different in that it's a 1-dose vaccine. The Pfizer vaccine is administered in 2 doses, 21 days apart, and the Moderna one is also 2 doses, 28 days apart. All vaccines are approved for ages 18 and older. The Pfizer vaccine has been approved for ages 16 and up. The Johnson & Johnson vaccine reports an 85% effectiveness right now. That's being continued to being looked at, and there's talk of perhaps a second dose about 2 months after the first dose, which may boost its effectiveness. The Johnson & Johnson vaccine uses AdVac technology, which is a genetically modified virus that carries the spike protein to the cell at a DNA level, where the mRNA vaccines, which are the Pfizer and the Moderna vaccines, create antibodies and an immune response is created. All 3 vaccines are administered intramuscularly, so they go into your muscle, and typically, as you see when you see on the news people being vaccinated, that goes right into your upper arm. Both the Pfizer and the Moderna vaccines use the messenger RNA to teach our cells how to make a protein, and that triggers an immune response inside our bodies, as opposed to traditional vaccines that may use a weakened or inactivated virus to create an immune response. Other vaccines that are in clinical trials are some by AstraZeneca and Novavax, and they're in their final stages of clinical trials. So hopefully they will be on the market soon also. All 3 current vaccines have begun clinical trials in children. It's a little bit different process that we'll talk about a little bit later in the presentation. One thing to note is on vaccines, if you are currently quarantining, so if you're person B, because you are identified as a close contact, you should delay your vaccination until your quarantine has ended per guidelines. So that would be one case where you'd want to wait the 14 days before you received your COVID vaccine, just to be sure you didn't actually develop actual COVID infection. So a little bit more on the mRNA vaccines, which is the Pfizer and the Moderna. These vaccines give instructions to our cells to make a harmless piece of the spike protein of the COVID virus. So this protein's on the surface of the virus. It's the little crowny spikes that you saw in one of the earlier slides. And then once these instructions are inside the immune cells, the cells use them to make the protein piece. After the protein piece is made, the cell breaks down the instructions and then gets rid of them. So it doesn't stay in your body. After that, the cell displays the protein piece on its surface, and our immune system recognizes that protein doesn't belong there, and it begins to build an immune response and make antibodies. So this is similar to what our body would do in a natural infection against COVID. At the end of the process, our bodies have learned how to protect us against the future COVID infections if it's seen in our body. The benefit of the mRNA vaccine, like all vaccines, is those vaccinated gain protection without ever having to risk the potential serious consequences of getting sick with COVID-19. So the Johnson & Johnson vaccine is a vector vaccine. This vaccine uses an adenovirus that has been altered so it can't reproduce. And in the case of the COVID vaccine, a gene with the SARS-CoV-2 spike protein is added to that viral strain. The virus is then the mode of transportation to get the spike protein into the cells, and it enters through the cells of the muscle system and the immune system via DNA. The adenovirus does not have the ability to change DNA in a person. Unlike the mRNA vaccines, it takes longer for an adenovirus-based vaccine to break down and be removed from the body. So this is one of the reasons that the Johnson & Johnson vaccine is given as a single dose at this time. So for our kids, as I mentioned, clinical trials have started. Pfizer and Moderna have vaccine clinical trials right now in 12 to 17-year-olds, and they're collecting their data. Pfizer has almost 3,000 kids in their clinical trial. So they have a nice pool to collect data in. The Moderna vaccine, they haven't really reached the numbers of enrollment as far as the same as Pfizer. They have about 800 kids right now. So they may be a little slower to continue their clinical trials and roll these vaccines out to younger kids. Johnson & Johnson trials, again, they're going to begin in the same age group and then move down to younger participants. Because the Johnson & Johnson vaccine is based on the vector vaccine, and these vaccines have been used in the past for things like Ebola, they do know that vector vaccines have been administered to children as young as 1 year of age in the past and were safe. So some of the things they need to consider when doing clinical trials for kids is that, smaller bodies, smaller frames may need a lesser dose. And children also have a more robust immune system than adults. So they may experience, an increase in side effects. So again, a lesser dose may be needed. Children make up about 25% of the US population. So while it is important for kids to be vaccinated, because of the way that children don't spread COVID, because they don't spread COVID the same way as adults, it's not necessary for kids to be vaccinated before schools open. Common side effects, if anyone out there has received their vaccine, most common side effects reported for one or both doses are pain or swelling at the site, fever, chills, headache, body aches, and fatigue. You cannot become sick with COVID from this vaccine. And the side effects that you're experiencing in your body, creating an immune response to the vaccine. After the vaccine, you can take Tylenol or Advil for discomfort or fever, rest, and drink plenty of fluids. So now you're vaccinated. You've made it through the side effects. You waited your 21 or 28 days, or you got your one jab. If you are receiving a 2-dose vaccine, make sure you get that second dose. And again, you won't be fully protected until about 2 weeks after that second dose. The Pfizer vaccine has so far been shown to be 95% effective at preventing laboratory-confirmed COVID-19 illness. And Moderna was 94.1% effective, the same. So right now, these vaccines are being recognized as preventing illness and therefore hopefully preventing transmission of illness. Right now, they're not saying that these provide immunity. It's something that's being looked at, and they'll continue to gather data. So I was asked the question, if I'm vaccinated, will I test positive for COVID? And the answer is no. And the reason for that is, again, the proteins created by the vaccines don't make their way to your respiratory tract. So when you're tested for COVID, either, we do the nasal swab. So those proteins would not show up in your nasal mucosa and be on the swab collected. If you were vaccinated if you got, say, your first COVID vaccine and tested positive a few days later, chances are you were exposed to COVID prior to your vaccine. After you're fully vaccinated, you can test positive for an antibody test to show that you have antibodies to the COVID vaccine. So again, like I said, this is from Johns Hopkins. Right now, everyone's very confident that the vaccines are highly effective at preventing illness. Data is being collected every day as more and more people are vaccinated. And hopefully, shortly we'll know if the vaccines provide immunity as well as reduce transmission. In Israel, early data shows that the viral load of people who got infected after vaccination was significantly lower than those who were not vaccinated. So that's a good thing. The lower your viral load, the less your chance of transmitting illness to someone else. So as we go on, we'll know whether vaccination will eliminate transmission of COVID by vaccinated individuals. If you were infected with coronavirus, it is safe to get a COVID vaccine after you've been infected. The CDC advises that those who have been diagnosed with COVID wait until after their 10-day isolation period is over. And if you were to have any symptoms that lasted longer than 10 days, you should wait until your symptoms are improved. Some people who may have compromised immune systems may have COVID symptoms up to 3 weeks after. After you have a COVID infection, you do have natural immunity. Right now, they are thinking that natural immunity probably lasts for a minimum of 90 days. So if you wanted to delay your COVID vaccine for that period, that would be fine too. You also would not have to quarantine within that 90 days of having the COVID infection. If, unfortunately, you were someone who became extremely ill with coronavirus and you received convalescent plasma, which is plasma that was donated by someone who previously had the COVID infection, you should wait the 90 days to start the COVID vaccine series after that. So things we have to look forward to. There are hundreds of treatments and vaccines in development and in trials throughout the world. I'm going to probably butcher the names of these drugs, but molnupiravir is an investigational antiviral that's being trialed in hospitalized and non-hospitalized patients with COVID in the United States. And this drug is an antiviral similar to if you were ever given Tamiflu. Tamiflu is for the influenza. Tamiflu is an antiviral. And what it does is it creates an error in the viral replication process. So it stops the COVID virus from replicating in your body. One thing that the studies so far have shown is a decrease in the time that someone who tests positive then tests negative. The second medication, favipiravir, is approved in Japan for influenza and in Russia for COVID. This drug inhibits an enzyme necessary for viral replication. In the US, it's being trialed to prevent severe disease in COVID patients and also for use in asymptomatic individuals who had direct exposure for post-exposure prophylaxis. So that would be a case where you're identified as person B, you were exposed to a COVID case. Clinical trials now, they're administering this drug to those patients who are person B to see if it prevents them from developing COVID-19. As I mentioned earlier, at-home testing is now available. Increased testing availability in schools, at home, testing sites, combined with the vaccines and drug therapies that are all being developed daily, are all going to work to reduce transition and overall cases of COVID-19. So we'd like to invite you all to join the PAC, which that's the Avon Grove Charter School version of herd immunity. So herd immunity or community immunity or PAC immunity happens when a sufficient portion of the population, typically greater than 75%, has immunity from a disease, either through prior infection or vaccination, and it makes the virus unable to spread. So it breaks the chain of transmission, less people get infected, and hopefully eventually the virus burns itself out. So right now, experts are estimating that herd immunity would require about 80 to 90% of the population to have COVID immunity, either naturally through a prior infection or through vaccination. So this is one of the reasons that we really are looking to do everything we can to get vaccinations out to everyone as soon as possible. And the more vaccinations that are approved under emergency use authorizations that get out into the market, the quicker we can get more people immunized. So this is just an example of herd immunity. So when you have no herd immunity, each infected person then transmits that infection to any particular number of people. And it varies by disease. So for COVID, when it was at its height, as far as infection, it was thought that one infected person could infect 5 other people. So then each of those 5 people would infect 5 more people, on and on and on. And that's how we got to pandemic status. A disease like measles was highly contagious when it was in its time. And one person infected with measles could infect, the thought was up to 15 people. So the cycle would continue. We caused mass outbreaks, but through natural immunity and vaccine immunity, we can break the transmission chain. And as you can see, once we have more people immunized or immune, the number of people that vaccine can be, or that virus can be transmitted to, lessens. There's just not a population out there that would be susceptible to it. So some of the benefits of herd immunity, in addition to stopping the spread of disease, is we can reopen the economy, reopen our schools, and then ease the burden on our medical system and the hospitals. So as more people are vaccinated and we continue to fight COVID-19, we can look to the future where we can throw our masks away, hug our loved ones, attend concerts and sporting events, and most importantly, reunite our AGCS PAC. And shameless shout out to myself for my Tim McGraw picture, because I'm looking forward to going back to Tim McGraw concerts. So I hope you guys all have something to look forward to. And if you have any questions regarding vaccines or COVID or what it's like in the school, if you're getting ready to send your kids back to school, please let me know. I'm happy to answer any questions. And because I'm nervous and talk fast, that's the end of my presentation.
Well, Holly, we thank you so much for your time and putting this presentation together, because we're all just flooded with information all the time. So it's nice to have somebody who knows what they're talking about break it down for us. So we really appreciate that. And I think you are a major factor in why our school has done so well up to this point in minimizing any sort of spread and keeping our school community safe. So thank you for that.
Thank you.
Yes. And like Holly said, if you have any questions for her specifically, you can email her anything at all, and she'll be glad to help you out. So thank you also to everyone who joined us tonight, and stay safe.